Hi, welcome to our channel of IGNOU audiobooks, Indira Gandhi National Open University, School of Continuing Education, SOCE, Master of Science, Food Nutrition, MSCDFSM, revised. First year. MFN 003 Food Microbiology and Safety. Unit-5 Food Hazards of Microbial Origin. 5.1 Introduction. Food is liable for contamination during the various stages of production to consumption. Cycle i.e. growth, harvesting, procuring, transporting or even during storage and distribution. The contaminant can be a physical agent, a chemical agent or a biological agent, as you may recall studying in Unit 2. The consumption of such contaminated foods is likely to cause adverse health effects to the consumers and hence referred to as food hazards. It is found that most of the food-borne diseases are due to microbiological contamination of the food. In recent years, a number of bacteria, viruses and parasites have emerged as food-borne pathogens which have resulted in numerous food-borne disease outbreaks. These outbreaks have had a major impact in terms of loss of human lives and economic costs. In this unit, we will try to understand about the food-borne diseases and their causative agents, how to control them and how to investigate and report a food-borne disease. Objectives. After studying this unit, you will be able to classify and describe the food-borne diseases, enumerate the various food-borne infections, intoxications and toxic infections, discuss about the causative agents and methods to control them, and report and investigate the food-borne diseases. 5.2 Food-borne diseases. Food-borne diseases are caused by the ingestion of foods containing toxic or infectious agents. In India, the diseases transmitted by food are commonly referred to as food poisoning. Food poisoning, in other words, is the term used to refer to the harmful effects of consuming food contaminated by microorganisms. Food-borne disease outbreaks in the community are of common occurrence, both in the developed and developing countries. A food-borne disease outbreak is defined as an incident in which two or more persons experience a similar illness, usually gastrointestinal, after the ingestion of a common food which is identified as the source of food-borne illness. You would be interested to know that more than 250 different food-borne diseases have been described. Most of these diseases are infections caused by a variety of bacteria, viruses and parasites that can be food-borne. Other diseases are poisonings caused by harmful toxins or chemicals that have contaminated the food, for example, poisonous mushrooms. Certain molds also produce toxins called mycotoxins in the food they attack. All these can lead to illness. Food-borne disease has been termed as the most widespread health problem in the contemporary world and an important cause of reduced economic prosperity. It is said that occurrence of a number of food-borne diseases is next only to common cold. In developing countries, the exact magnitude of the problem of food-borne diseases is not fully recognized. It is estimated by the World Health Organization, WHO, that the ratio of actual to reported cases of food-borne diseases varies from 25:1 to 100:1. In India, the food-borne diseases are rarely recorded, and when recorded, most often they are categorized under gastroenteritis, inflammation of the stomach and small and large intestines. As the effects of food-borne diseases are mild and relatively of short duration, the affected persons ignore them and are not aware of the linkage of their Illness to the foods consumed. On most occasions, they do not take medical help. Even if they seek medical aid, it will be on an individual basis, except in rare cases, where a large number of people are affected simultaneously. How then can we recognize a food-borne disease? What are its usual symptoms? Food-borne diseases generally involves a disturbance of the gastrointestinal tract with abdominal pain, diarrhea and sometimes vomiting. Symptoms of food-borne illness range from mild gastroenteritis to life-threatening neurologic, hepatic and renal syndromes. Next, what are the different types of food-borne diseases? What are the factors responsible for the outbreak of these diseases? In the subsequent subsection, we will learn about these crucial aspects. 5.2.1 Types of food-borne diseases. You already know that food-borne diseases or food poisoning is a condition resulting from eating contaminated food. The disease-causing culprits are the microorganisms or pathogens including fungi, bacteria, parasite or virus. You would realize that some pathogens produce toxins in food which when consumed can cause illness. On the other hand, pathogens may be present in food which when consumed may produce a toxin in the gut or invade and destroy the healthy tissues. Accordingly, food-borne diseases are classified into three categories, namely I. Food intoxications, 2. Food infections, and 3. Food-borne toxic infections. Food infection generally involves microorganisms present in the food at the time it is consumed. Once inside the human body, they begin to grow and cause disease. Salmonellosis, caused by the bacteria Salmonella, commonly associated with chicken or egg, is an example of food-borne infection. Food intoxication, on the other hand, involves toxic substances produced in the food by microorganisms before it is consumed. The toxin present in the food makes the person feel sick. Staphylococcus aureus food-borne disease is an example of illness that results from the consumption of toxins in food. The third category, i.e., food-borne toxic infections, are caused by the ingestion of a large number of enterotoxigenic strains of bacteria which, while multiplying in the intestine, produce and release enterotoxins in the intestines. Apart from the three categories given herewith, you would come across certain molds and fungus which produce toxins in the food they attack. The toxins produced by the molds are called mycotoxins. Poisoning caused by ingestion of a food or feed that contains a mycotoxin is called mycotoxicosis. Examples include aflatoxins found in peanuts and sometimes in maize, rice and sorghum. Ergot fungus is associated with bajra, jowar and rye. This fungus produces toxins which result in ergotism. In addition, there are a few naturally occurring toxins in food which can cause illness. We shall study in details about these food-borne diseases in the next subsections. Starting with food intoxicants. 5.3 Food-borne intoxications. Food-borne intoxications or poisonings are caused by either the ingestion of toxicants that are found in the tissues of certain plants or animals, or 2. Toxins formed and excreted by bacteria and fungi while they multiply on or in foods, as well as toxins formed and excreted by algae and ingested and concentrated by shellfish during their growth in seawater, or 3. Poisonous substances that may be intentionally or incidentally added to foods during producing, processing, transporting or storing. The most important bacterial food-borne intoxications are A. Staphylococcal poisoning, B. Bacillus cereus poisoning, and C. Botulism. Let us learn about these intoxications. 5.3.1 Staphylococcal poisoning. Staphylococcal food poisoning is one of the most frequently reported foodborne diseases which is common throughout the world. The disease is caused by the ingestion of the enterotoxin formed in food during the growth of certain strains of Staphylococcus aureus as they multiply in protein-rich foods. The toxin is referred to as enterotoxin because it is a toxin produced by bacteria that is specific for intestinal cells and causes gastroenteritis, i.e., inflammation of the lining of the intestinal tract. Staphylococcus aureus is a small, 0.5 to 1.0 millimeters in diameter, gram-positive, non-motile, non-spore-forming coccus or a spherical cell which divides to form irregular clusters of cells resembling bunches of grapes as you can see in the figure 5.1. It is aerobic and facultative anaerobic with an optimum growth temperature around 35 degrees Celsius to 37 degrees Celsius and is capable of growing within the temperature range of 7 degrees Celsius to 48 degrees Celsius. Under optimal conditions, the organism doubles every 20 minutes after 1 hour lag. Staphylococcus aureus can tolerate foods containing 15% to 20% salt as it has a low water activity (AU) level 0.86. Enterotoxin produced by it are designated as A, B, C1, C2D and E. The commonly identified toxins involved in the outbreaks are A and D. The toxins can be detected only during the late exponential or stationary phase of growth. Small amounts of toxin, about 1 gram, can cause the illness in sensitive persons. The toxins are heat stable in their crude form. They are able to withstand boiling temperatures in a food for several minutes up to 30 minutes. A number of commercial sterilization processes such as normal cooling process, spray drying and pasteurization have found not to inactivate the toxin. The organism itself is not heat resistant and will be destroyed by pasteurization. After the ingestion of a contaminated food, the symptoms appear within 1 to 6 hours, and in most cases, between 2 to 4 hours. The period is dependent on the dose ingested. Greater the enterotoxin ingested, shorter is the time for appearance of the symptoms. Onset is heralded by nausea followed by vomiting and abdominal cramping. Other symptoms may include fever and chills, weakness and headache, but do not last for long, usually from less than 1 day to 2 days. Mortality in the case of Staphylococcus aureus toxin is low, but a few fatal cases have been recorded in the children and elderly. The treatment for poisoning is usually symptomatic, which includes oral or intravenous administration of fluids. The most important source of Staphylococcus aureus is man. The principal reservoir is the nose, followed by hair. It is harbored on human skin, especially on cuts, burns, boils, etc., which can cause contamination of foods. So its presence in cooked or processed foods can serve to indicate poor hygiene among food handlers. The foods involved in S. aureus food poisoning are typically those that have been handled and then temperature abused prior to consumption. The common foods implicated in the outbreaks are raw milk, raw meat, custard, cream, bakery foods, poultry and ham, egg foods, fermented meat and dairy products. In India, the dairy product khoya, the major sweet base, has been involved in several outbreaks. Animals can also act as a source of S. aureus, the udder and teat canal in cows and buffaloes are the common sites of the organism. It is not possible to totally eliminate the staphylococci out of foods as they are present almost everywhere. Therefore, only preventive measures have to be undertaken to minimize the risk by inhibition of staphylococci from multiplying and producing the toxin. Some strains of S. aureus are capable of producing heat-stable toxins in food. It is this toxin that causes the typical symptoms associated with food poisoning, which we had studied earlier. So let us now look at some of the measures which could decrease the risk of infection. These are: 1. Keep the handling of cooked foods to a minimum. 2. Once the food is prepared, it should be held at temperatures above 56 degrees Celsius. 3. Cooling of foods rapidly and storing chilled foods in shallow containers at temperatures below 7 degrees Celsius. 4. Processing of foods within a time span of 1 to 2 hours, in which staphylococci are in the lag phase. 5. Minimizing the cross-contamination from raw to cooked foods and also by taking precautions to avoid contamination from working surfaces, equipments, and utensils. 6. The personnel handling the foods have to take good personal care adopting hygienic practices. They should wear disposable gloves wherever possible. With this we come to end of our discussion about staphylococcal poisoning. Next, we shall get to know about Bacillus cereus poisoning. 5.3.2 Bacillus cereus poisoning. Bacillus cereus poisoning is caused by the organism which is a large, 3 to 5 meters long and 1 to 4 meters wide, gram-positive, motile and spore-forming rod, as shown in the Figure 5.2. B. Cereus is an aerobic organism but is also capable of growing under anaerobic conditions. Mesophilic strains of B. cereus grow between temperatures of 10 to 45 degrees Celsius, with an optimum growth temperature range being 20 to 35 degrees Celsius. The psychrotrophic strains, however, grow and produce toxins at temperatures as low as 4 degrees Celsius. Further, since it is a spore former, the heat-resistant spores can survive the cooking process. Hence, it requires a severe heat process to destroy the spores. Bacillus cereus can cause two distinctive forms of food poisoning, which differ both in their incubation time and the symptoms they elicit in the victim. Some of the strains produce and release a toxin which causes diarrhea, while some other strains produce an emetic toxin in the intestine which causes nausea and vomiting in the person consuming food containing the toxin 2 to 5 hours after food ingestion. The diarrheagenic toxin is relatively heat-sensitive and is destroyed in 30 minutes at 56 degrees Celsius and in 20 minutes at 60 degrees Celsius. The emetic toxin, however, is capable of tolerating high temperatures, 126 degrees Celsius, for 90 minutes. The vomiting, emetic type of illness exhibits the symptoms in a shorter time, usually between 1 to 5 hours and occasionally anywhere between 15 minutes to 11 hours. The symptoms observed are general malaise, acute attack of nausea and vomiting. Diarrhea is also observed in some cases. The other form of illness is characterized by acute abdominal pain, profuse watery diarrhea, nausea, vomiting, fever, chills and body aches. The incubation period is anywhere between 8 to 16 hours and lasts for less than 24 hours. The treatment for the B. cereus Food poisoning is symptomatic. Being ubiquitous in nature, B. Cereus is commonly found in soil and in vegetables and grains. The prepared foods, which are the vehicles of diarrheal form of illness, include cereal dishes, puddings, milk and dairy products, mashed potatoes, sauces, vegetable and chicken soups and meat and rice dishes. The usage of spices in preparation of meat dishes adds the B. Cereus spores to the dishes, as spices harbor spore-bearing bacilli. If the preparations like mutton and chicken biryani are not adequately heated, it allows the spores to germinate during the subsequent holding of the prepared food at room temperature for longer periods. This leads to a heavy growth of the vegetative cells. The other type of illness, the vomiting, emetic type, results from the consumption of fried rice. The rice is usually prepared in large quantities and allowed to cool at the room temperature, which is an ideal condition for the germination of surviving spores. The rice is fried just before serving. The heating process generally reduces the bacteria, but depending on the microbial load, the person consuming the food may get affected. B. Cereus is normally found on or in the foods. There is a possibility that the spores produced by the psychotrophic strains survive the cooking temperatures and germinate at room temperatures. Therefore, prevention of illness through foods can be minimized by following certain precautionary measures, which include I. The rice cooked for preparation of fried rice should be boiled in small quantities whenever required, depending on the customer inflow, which helps in reducing the holding period of cooked rice. 2. If the rice has to be cooked in large portions, care should be taken to ensure that the hot rice is held at temperature not less than 63 degrees Celsius. For any reason, if this is not possible, the cooled food should be divided into small portions, cooled quickly in shallow containers and transferred to the refrigerator and held at temperature less than 7 degrees Celsius. This should be carried out within 2 hours of cooking. 3. Always avoid holding the cooked rice at room temperature. This is highly essential in avoiding B. Cereus food poisoning. Having studied about Bacillus cereus poisoning, finally we move into the third food borne intoxication, i.e. botulism. 5.3.3 Botulism. Botulism is an extremely serious neurological illness. It is caused by the ingestion of improperly preserved canned foods containing a neurotoxin produced by Clostridium botulinum. This is generally a fatal disease and carries a high risk of mortality, 35–40%, although its occurrence is rare in India. The Clostridium botulinum is an obligate, anaerobic, mesophilic, motile and gram-positive rod, 4 to 8 meters by 0.9 to 1.2 meters, which produces a heat-stable spore. Some strains of C. botulinum are psychotrophic, which are capable of slow growth and produce toxin at low temperatures, 3.3 degrees Celsius. There are 8 types of toxins produced by it and are designated as A, B, C1, C2, D, E, F and G. All the strains do not produce single toxins, as some produce mixed toxins like C1, which produces C2 toxin, but also some D toxins. Most human cases of botulism have been reported to be caused by A, B and E toxins. The toxins are called neurotoxins as they act upon peripheral nerves of the involuntary muscles of the body. The toxins A and B can be inactivated by heat treatment at 80 degrees Celsius for 10 minutes, while the E toxin can be destroyed by heating for 5 minutes at 60 degrees Celsius. The incubation period of the disease ranges from 2 hours to 14 days after the ingestion of contaminated food. The first symptoms appear between 18 to 36 hours, and the signs and symptoms depend upon the type of toxin. Generally, the symptoms are nausea and vomiting, occasionally diarrhea, headache, dizziness, and persistent constipation, followed by blurred vision. Clostridium botulinum is widely distributed in soil and marine sediments throughout the world. It is also found in the intestinal tract of animals, including fish. Most of the outbreaks of botulism have been associated with the products of fish or marine animals, meat, fruits, and vegetables, including mushrooms. Insufficiently heated canned and bottled foods are at a high risk, as these provide the anaerobic, without air, environment required by the organism to grow. The types of foods which have acted as vehicles of infection differ from country to country. Generally, the toxins A and B have been implicated in improperly home canned foods. The type E outbreaks have occurred by the ingestion of fermented or smoked marine products, fruits, and vegetables, including mushrooms. Home cured ham and other meat products have been implicated in the outbreaks reported from Western Europe. The organism can be isolated occasionally from many foods because of its widespread occurrence in the environment. However, it is interesting to note that because of their non-proteolytic characteristics, their growth in foods cannot be detected by off odors and off flavors. Botulinum produces preformed toxins. Remember, it is the ingestion of the preformed toxin, which is the most potent natural toxin known to man, that causes the illness known as botulism. Ingestion of live organisms of C. Botulinum is not the cause for foodborne botulism. The toxin can be destroyed by heat treatment, 800 degrees Celsius or above, for only a few seconds. The botulism can be prevented by killing C. botulinum spores in foods through following these steps: I. Processing. 2. Eliminating the recontamination of processed foods. 3. Destroying the toxin by proper heating of the processed food before serving. 4. Proper storage. Discarding the product that has developed signs of spoilage, e.g., of odor, bulging cans, and gas bubbles on opening the can. And 6. Avoiding tasting of suspected food unless it is heated to 100 degrees Celsius. A heat process called botulinum cook is commonly recommended for low-acid canned products. In conclusion, botulism is an extremely serious foodborne disease, and unless recognized and treated promptly, it carries a high risk of mortality, 35 to 40%. It is the most severe form of food poisoning. 5.4 Foodborne infections. Foodborne infections, as you may recall reading earlier, are caused by the ingestion of pathogenic microorganisms that penetrate the intestinal mucosa and multiply or migrate into other tissues where they multiply. Common foodborne infections include Salmonellosis, shigellosis, Vibrio parahemolyticus, gastroenteritis, E. coli, diarrhea, infective hepatitis, etc. Let us study about a few of these infections. 5.4.1 Salmonellosis. Salmonellosis is a common foodborne disease all over the world. There are approximately 1,600 different strains, serotypes, of Salmonella. The most common of The species is Salmonella typhimurium, which causes typhoid. Figure 5.4 illustrates the Salmonella organism. The members of the genus Salmonella are short, 1 to 2 meters, motile, gram-negative and non-sporing rods. They are aerobic and facultative anaerobic. Several of the serotypes have been named after the place from where they were first isolated, like S. Newport, etc. These organisms grow best at around 38 degrees Celsius, however, the growth is slow at temperatures below 10 degrees Celsius. They are heat sensitive and are destroyed by pasteurization and heat treatments at 60 degrees Celsius in 15 to 20 minutes. Salmonellosis occurs following the ingestion of viable cells of the organisms and is the most common type of food-borne illness. The occurrence of the disease is largely dependent on the resistance of the individual, the strain involved, and the number of bacteria ingested, which varies. S. enterotides and S. typhimurium are the frequently implicated organisms in cases of salmonellosis. Other serotypes have also been involved in food poisoning from strain to strain. The incubation period varies from 6 hours to 3 days but is normally between 12 to 36 hours. The symptoms are nausea, diarrhea, abdominal pain, vomiting and fever. In some cases, this may be preceded by chills and headache. Although death from the disease is rare, it can occur in at-risk groups, e.g. infants, elderly, and the immunocompromised. The illness usually lasts for several days. The prime source for salmonella is the animal, but human beings are also indirectly the source after once becoming infested and are known as carriers. The domestic animal food sources like chicken, pigs, turkey, etc. are the main reservoirs of the organisms. The organisms spread from carcass to carcass at the slaughterhouse. The types of foods involved in food-borne salmonellosis have been wide-ranging and involve mainly cereal and grain products, desiccated coconut, chocolate and dairy products. The egg and egg products, which have not been pasteurized, were implicated in several outbreaks. Meat and meat products also are the vehicles of food-borne disease outbreaks. Milk and milk products like fermented milks, ice cream, cheese, etc. have also been responsible for diseases. Foods such as desiccated coconut, chocolate and dairy products are also involved in food-borne diseases. In kitchen, the foods may get cross-contaminated by the equipments and utensils. The prevention of salmonellosis involves adopting several precautionary steps which include I. Checking of the animal to be free from contamination. 2. Avoiding cross-contamination from raw to cooked foods. 3. Destruction of salmonella by proper cooking. 4. Cooling the prepared foods rapidly to below 7 degrees Celsius and storing within the refrigerators or freezers or held at a temperature above 60 degrees Celsius. Removing food handlers who are carriers from the food preparation area. And 6. Improving general sanitary conditions of the premises and ensuring better pest control. Next, let us get to know shigellosis, the food-borne infection caused by Shigella. 5.4.2 Shigellosis, bacillary dysentery. Shigellosis is caused by the genus Shigella and is a common enteric intestinal infection. The genus has 4 serological groups, of which S. SOCE, subgroup D, and S. flexneri, subgroup B, are the important ones. The Shigella are non-motile, aerobic and facultative anaerobic, non-sporulating gram-negative rods in the family. Enterobacteriaceae. The optimum temperature for the growth is 37 degrees Celsius with a temperature range of 10 to 40 degrees Celsius. They can withstand salt concentrations of up to 6% and are heat sensitive. They utilize glucose and other carbohydrates and are inhibited by potassium cyanide. The number of organisms to cause illness is low and the main mode of transmission is from human to human. The incubation period depends upon the organism, the affected person's age, and the health status. The incubation period is usually short, ranging from 1 to 7 days, usually 1 to 3 days. The symptoms are diarrhea, accompanied by fever, nausea, and sometimes vomiting and cramps. The stools of the affected person may contain blood, mucus, and pus. Although shigellosis is usually transmitted by contaminated water, where food has been involved, the vehicles of infection are milk, shellfish, raw vegetables, Mexican dishes, and various salad preparations. The infection is transmitted by the food handler with poor personal hygiene and acts as a carrier. The food handlers contaminate the food or food preparation surfaces. The following measures help to minimize the problem. The food handler, who is the primary carrier, has to be educated regarding safe handling of foods and made to follow strict personal hygiene practices like washing hands after visiting the toilet. 2. The leftover foods have to be chilled rapidly or held at temperatures at which Shigella cannot grow above 40 degrees Celsius. 3. Cooking and reheating of foods to safe temperatures is also essential for preventing shigellosis. From the discussion above, it is evident that food handlers with poor personal hygiene are one of the main causative agents of shigellosis. Next, let us get to know about the outbreaks of illnesses associated with Vibrio parahemolyticus. 5.4.3 Vibrio parahemolyticus gastroenteritis. Outbreaks of illness associated with Vibrio parahemolyticus are frequently reported from Japan. V. parahemolyticus associated gastroenteritis is the name of the infection caused by this organism. The organism is a facultative anaerobe, straight or slightly curved, motile, gram-negative rod with a single polar flagellum as illustrated in Figure 5.6. It is also slightly halophilic, salt lover, and can grow in 8% NaCl but grows ideally between 2 to 4%. The optimum temperature at which it can grow is 35 to 37 degrees Celsius but exhibits growth trends over a wide range of temperatures from 8 degrees Celsius to 44 degrees Celsius. In the V. parahemolyticus gastroenteritis, the symptoms are severe abdominal pain, vomiting and diarrhea, leading to dehydration and fever. The incubation period is usually from 12 to 24 hours and the illness may last from 1 to 7 days. V. parahemolyticus is found in marine environment. The organism is found abundant during the warm months and it colonizes the water and marine life. Fish and shellfish are the two major sources of contamination and are generally implicated in the outbreaks. The important cause of the outbreaks in Japan is the habit of consuming raw fish as such, hence there is a high prevalence of the disease in the country. In India, the disease has been reported from Kolkata. The major reservoirs of the pathogenic bacteria are crabs, shrimps and certain freshwater fish commonly consumed in West Bengal. Normally, the recovery after illness takes place anywhere between a few hours to 10 days. The organism is easily destroyed by heat. The best way to, however, prevent the illness due to whey parahemolyticus is by I. Thorough cooking of the seafoods. 2. Avoiding cross-contamination of cooked food from raw foods and 3. Proper refrigeration of raw seafoods. 5.4.4 Enteropathogenic Escherichia coli diarrhea. Escherichia coli, E. coli, is an important organism as it is regarded as a part of normal intestinal flora of humans and animals. Its presence in food has been used as an indicator for fecal contamination. The E. coli is a short, non-spore forming, facultative anaerobe, motile and gram-negative rod. Some strains of E. coli can cause acute gastroenteritis affecting children. Such strains are designated as enteropathogenic E. coli, EPEC. Diarrheal disease due to EPEC is age-related, usually occurring in infants. Outbreaks involving babies, even fetuses, have been reported from hospitals and nurseries. There are certain serotypes of E. coli, known as verotoxigenic E. coli, VTEC, which may cause diarrheal disease or more serious forms of illness, such as acute renal failure or long-term kidney problems. VTEC produces toxins referred to as verotoxins. Because they affect vero cells, a tissue culture line of monkeys' kidney cells. The main serotype of verotoxin-producing E. coli is E. coli 0157. This strain of E. coli produces a powerful toxin and can cause severe illness. The optimal temperature for the growth of E. coli is 37 degrees Celsius, but it can grow between 7 to 43 degrees Celsius. The organism is heat sensitive. The pasteurization and normal cooking temperatures are effective in destroying the organism. The organism survives well at low temperatures with the incubation period between 12 to 72 hours. The symptoms include abdominal pain, diarrhea, vomiting and fever. Blood and mucus in stools may also be observed. EPEC may be present in poultry, beef products, pork, lamb, apple cider, meat and dairy products, like cheese, which has been implicated in some outbreaks reported from USA and UK. It is spread through fecal contamination of food, water and from person-to-person transmission. Diagnosis of its outbreak is only possible through culture and identification of the germ using biochemistry and serology. The prevention of EPEC-induced gastroenteritis is to adopt strict personal hygiene and good sanitary practices. Next, let us get to know about infectious hepatitis. 5.4.5 Hepatitis A. Infective hepatitis, which is caused by hepatitis A, is a viral disease which spreads through food. How does the hepatitis A virus look like? Hepatitis A virus is presumed to replicate initially in the gastrointestinal tract and then it spreads primarily to liver, where it infects hepatocytes, liver cells, and Kupffer cells. Infective hepatitis is a major public health problem all over the world. The major source of the virus has been man. The disease is transmitted through the oral fecal route. The sources of outbreaks involving food have been reported, but the long incubation period of 15 to 50 days, normally between 28 to 30 days, makes it difficult to investigate. However, foods such as ice, water, ice cream, milk, pastries, salads, sandwiches, shellfish, Raw foods or foods subjected to additional handling after cooking are the major food vehicles for virus transmission. The typical symptom of the disease is jaundice accompanied by abdominal pain, headache, fatigue, fever, etc. Jaundice, as you may already know, is the symptom characterized by yellowish discoloration of the whites of the eyes, skin, and mucus membranes caused by the deposition of bile salts in these tissues. Shellfish from polluted waters, fruits and vegetables contaminated by feces and various salads prepared under unhygienic conditions were responsible for several outbreaks. The best way for the prevention of infective hepatitis is to ensure good personal hygiene of food handlers like proper hand washing, etc. The other approach is to avoid foods known to have been procured from polluted water sources. Finally, let us get to know about shellfish poisoning. 5.4.6 Shellfish poisoning. Shellfish like oysters, mussels and clams are generally bred in the sewage polluted beds or brackish water. The shellfish poisoning in humans is caused due to the consumption of oysters, mussels and clams, etc., which accumulate the toxins produced by dinoflagellate algae Gonulax catenella. The mussels and clams concentrate the dinoflagellates by filtering them as food from the surrounding water. If the concentration exceeds 200 cells per ml in the surrounding water during a bloom, they will contain sufficient toxin to cause illness to those consuming the shellfish. The shellfish poisoning assumes importance due to the fact that they are consumed undercooked or uncooked. They also harbor other pathogenic microorganisms. In India, shellfish poisoning has been reported from the coastal areas of Tamil Nadu and Karnataka. Our discussion so far focused on food intoxications and food infections, which you learned earlier are the types of food-borne disorders. The third category of food-borne disorders are toxic infections. Let us get to know about these disorders next. 5.5 Food-borne toxic infections. The food-borne toxic infections, you may recall reading earlier, are caused by the ingestion of a large numbers of enterotoxigenic strains of bacteria which, while multiplying in the intestine, produce and release enterotoxins in the intestines. Few of the foodborne toxic infections include Clostridium perfringens gastroenteritis, enterotoxigenic Escherichia coli gastroenteritis, cholera, listeriosis, etc. Now we will focus on these major foodborne infections and study about the characteristic features of the organism responsible for these infections. 5.5.1 Clostridium perfringens gastroenteritis. Clostridium perfringens associated gastroenteritis is a food-borne disease which is frequently reported and is a common cause of illness. The organism Clostridium perfringens is a large, 2 to 8 meters long, 1 meter wide, non-motile, gram-positive, obligate, anaerobe and spore-forming rod. The organism does not produce spores while growing in the food, but it forms spores when it reaches the intestinal tract. The spores of C. perfringens can withstand boiling temperatures for up to 6 hours. The optimum temperature for growth is between 43 degrees Celsius to 45 degrees Celsius, whereas the maximal temperature at which it can grow is 55 degrees Celsius. Its growth gets restricted at the temperatures of about 15 to 20 degrees Celsius. The vegetative cells of the organism are usually destroyed at the temperatures of 60 degrees Celsius and above. The strains of C. perfringens are classified into 5 types A to E, depending on the type of toxin produced. The strains of C. perfringens type A, the spores of which are heat resistant, survive at temperatures from 95 to 100 degrees Celsius for periods of up to 1 hour. These are responsible for several outbreaks in the United Kingdom. The incubation period for the disease ranges between 8 to 22 hours, but normally the symptoms occur between 8 to 12 hours after the ingestion of contaminated food. The important symptoms are severe abdominal cramps and watery diarrhea. Vomiting is rare. The recovery is usually within 1 or 2 days. The C. perfringens is the most common of all pathogenic bacteria, as it is widely distributed and usually can be isolated from soil, dust, marine sediments, human feces, and animal manure. They are also found in raw meat and poultry. The usual vehicles of infection are the dishes prepared from meat and poultry. The outbreaks have been reported from places where a large number of people eat, like the restaurants, institutional canteens, hospitals, etc. The major reason for the cause of outbreaks is the time temperature abuse after the preparation of the dishes. The slow cooling process, which sets in while the cooked food is held at room temperature, aids in the growth of spore germination, and a large number of vegetative cells may develop within a few hours. C. perfringens causes a mild but common type of food poisoning. Its spores are heat resistant, surviving normal cooking. The organism usually forms the toxin once it is in the human intestine and has started to sporulate. The toxin formed in food is not very heat resistant, destroyed by heating at 60 degrees Celsius for 10 minutes. C. perfringens food poisoning causes severe abdominal pain and prolific diarrhea. Sometimes fever, nausea, and even vomiting. Recognizing the debilitating effects of the illness, various regulations, as well as the maximum permissible limits for aflatoxins have been established for a range of commodities by the government. Next of the mycotoxins highlighted in this section is deoxynivalenol. Let us get to know about this toxin. 5.6.2 Deoxynivalenol mycotoxicosis. During 1987, an outbreak estimated to have affected over 50,000 people in the state of Jammu and Kashmir was traced to the consumption of preparations made out of wheat products like wheat flour and refined wheat flour, barley, oats, rye, maize, sorghum, and rice. The disease symptoms included pain in the stomach 10 to 15 minutes after consuming food and vomiting. The problem was diagnosed as deoxynivalenol mycotoxicosis. What caused the illness? It was found that unseasonal rains at the time of harvest of wheat had resulted in the damage to wheat by fungi like fusarium and the production of deoxynivalenol and other mycotoxins, known as trichothecenes, caused the outbreak. The mycotoxins can have effects on the immune system as well. It is thermally stable, partially, so once formed, it is likely to persist through storage and food chain. The only good thing is that as the toxin is water-soluble, a significant proportion can be removed by washing grain. Hence, preventive measures rest on advocating washing of grains before use. 5.6.3 Ergotism. Ergot is a disease of cereal crops like rye, wheat, millet, sorghum, and grasses. Ergot of rye is a plant disease caused by the fungus Claviceps purpurae as highlighted in Figure 5.17. The infected flower, instead of developing into a normal grain, develops to a dark black mass. The ergot Grain gets harvested along with good grains and contaminate the rye crop. Poisoning, attributed to ergot of rye, is, therefore, referred to as ergotism. This disease was common in Europe in Middle Ages. In India, ergotism is caused by the consumption of bajra. Contaminated ergot was caused by the fungus Claviceps fusiformis. The ergotism found in India was manifested as nausea, vomiting, giddiness, and prolonged sleepiness and is termed as enterogotism, while the European classical ergotism was characterized by convulsions and gangrene. In India, several outbreaks due to the consumption of ergoti bajra were reported from Maharashtra, Gujarat and Rajasthan. With our discussion on ergotism, we come to an end of our study on mycotoxins. So far we have studied about food-borne hazards of microbial origin. We learned that microorganisms, bacteria, fungus themselves, or the toxins produced by them in the food or in the human body can lead to food-borne diseases. You would be interested to know that other than microorganisms and their toxins, certain naturally occurring toxins in foods can also cause food poisoning. We shall learn about these naturally occurring toxins and the diseases caused by them in the next section. 5.7 Food-borne diseases due to naturally occurring toxicants. A variety of naturally occurring food toxins have been linked to human ill health and death. Among them, the principal toxicants that have been responsible to cause disease outbreaks in humans in India include the following: I. Lathyrism. 2. Veno-occlusive disease. 3. Epidemic dropsy. Let us discuss each of these. 5.7.1 Lathyrism. Human lathyrism is endemic in some parts of the country, especially certain areas of Madhya Pradesh. Cases of lathyrism have also been known to occur in Uttar Pradesh, Karnataka, Maharashtra, and Andhra Pradesh. Several reports of human lathyrism have been recorded in the past. The disease has been attributed to the consumption of Lathyrus sativus, popularly known as kesri dal, as a staple diet. The plant and seeds of Lathyrus sativus. Why should consumption of kesri dal lead to lathyrism? A neurotoxic amino acid, oxalylaminoalanine, BOA, has been identified as the constituent of the kesri dal responsible for the disease. Kesri dal or Lathyrus sativus is a drought-tolerant legume. It actually flourishes in conditions of the most devastating flood and drought when no other food crop survives. The grains contain 20 to 32% protein, but they also contain a number of antinutritional compounds known as lathyrogens. Lathyrogens are the neurotoxic amino acids that act as metabolic antagonists of glutamic acid, a neurotransmitter in brain. BOA, found in kesri dal, is a neurotoxic amino acid consumed in large amounts. During times of drought, a toxic syndrome, lathyrism, develops, resulting in a crippling paralysis of the lower limbs. The symptoms at the time of the onset include heaviness and stiffness of the limbs, muscle cramps, tremors, and involuntary movement of the upper extremity. However, it is encouraging to note that a declining trend of lathyrism has been observed in our country. This trend is attributed to the fact that the consumption of L. sativus in the endemic areas has come down during recent years, mainly because of availability of alternative grains like wheat. Further, increased cost of L. sativus and the ban on sale of L. sativus under the PFA Act has contributed to the declining trend of lathyrism. 5.7.2 Veno-occlusive disease, VOD. Veno-occlusive disease, VOD, of the liver is a well-recognized form of toxic liver injury produced by pyrrolizidine group of alkaloids. The VOD outbreak was observed in the tribal areas of Sarguja district of Madhya Pradesh and was characterized by ascites, pain in the epigastrium and death. This incident occurred due to the consumption of minor millet, Gondhali panicum milia contaminated with toxic wheat seeds of Crotalaria nanoburn. The seeds of Crotalaria contain monocrotaline and other toxic alkaloids belonging to the pyrrolizidine group. 5.7.3 Epidemic dropsy. There are several reports about outbreaks of epidemic dropsy in the last 3 decades. In the country, epidemic dropsy was first reported from Kolkata in 1877 and the most recent outbreak in Shivpuri district in Madhya Pradesh. The disease results from the ingestion of mustard oil or other vegetable oils contaminated with Ajmon mexicana seed oil, whose seeds closely resemble the mustard seeds. Figure 5.20 illustrates the picture of Ajmon mexicana. The disease is characterized by oedema over ankles, gastrointestinal disturbances, vascular changes, ocular changes and cardiac insufficiency. Ajmon toxicity presumably is due to the toxic alkaloid, sanguinarine, which interferes with the oxidation of pyruvic acid, accumulates in blood resulting in signs and symptoms. Mortality is variable but it disables the victim for a long time. Epidemic dropsy has been a recurrent problem in those areas where mustard oil is used as a cooking medium. In other parts of the country, the disease is confined to the communities using mustard oil or deliberate adulteration of other vegetable oils with Ajmon seed oil by traders. With our discussion on naturally occurring toxins, we come to an end on our study of food-borne diseases. You would agree it was an exhaustive reading. Before you relax, we suggest you look up the exercises given herewith under Check Your Progress. Exercise 4. Answering these questions will help you review your understanding on the topic. The information above provided you the basic knowledge on food-borne diseases. With this knowledge, we hope you would be able to identify the food-borne diseases. In the field setting, reporting and investigation of food-borne diseases, you would realize, is very crucial for its prevention. In the next section, we have focused on this aspect and you will find a detailed discussion on the basic elements of outbreak. Investigation. 5.8 Reporting and investigations of food-borne diseases. We have already seen earlier in this unit that the reporting of food-borne diseases is very little due to various lapses. You would now realize how crucial it is to record such instances for further investigations. Reporting an outbreak incidents could help in identifying the mode of transmission, the causative organism, and in facilitating necessary action to be undertaken. Further, it helps in formulating preventive strategies for the future so as to avoid or minimize the chances of occurrence of an outbreak. Hence, it can be said that the epidemiological surveillance of food-borne disease is fundamental to the planning and management of food safety program in a country. One of the important components of the surveillance is the reporting of a disease as it occurs, which would enable the public health personnel to take the necessary actions. It is very difficult to have information on every food-borne disease that occurs in a country. In the Indian context, the information on the occurrence of a food-borne disease is mostly through the newspaper reports and especially in vernacular press. In spite of the best communication and networking facilities, the ratio between actual to reported cases is 25, 1 in developed countries, whereas it may be as low as 101 in developing countries like India. We have seen earlier also that in India, the food-borne diseases are rarely recorded. And when recorded, most often they are categorized under gastroenteritis. Under Indian food regulations, all the physicians working in an area have to inform the occurrence of a food-borne disease to the medical officer designated by the government. However, this rule is rarely implemented and the food-borne diseases are not recorded systematically. What is the process of recording or investigation of an outbreak? Are you aware of the process? Let us learn about the procedure in the next section. Investigation of an outbreak. Once a suspected outbreak of a food-borne disease has been reported, the basic elements of an outbreak investigation include 1. Receipt of an initial data. 2. Determination of whether an outbreak has occurred. 3. Description of cases in terms of time, place and person. 4. Epidemiological investigation. 5. Formulation of hypothesis. 6. Laboratory studies. And 7. Synthesis of findings with conclusions and recommendations. You would realize that these elements are a part of an exercise of outbreak investigation. And these steps are discussed herewith. A. Receipt of initial data and assessment. How does an outbreak gets noticed? Food-borne diseases are often brought to the attention by admission of patients with symptoms of vomiting and diarrhea. The initial step, therefore, in any investigation is to verify the diagnosis or to confirm that illness has occurred. This is followed by an assessment of whether there is an outbreak, and if so, its potential magnitude and importance. The major factor to be considered in food-borne disease is that a true cluster of cases of the same illness is present. Certain additional factors which need to be considered include 1. The number of people possibly affected. 2. The severity of the suspected disease. 3. Time for which illness occurred. And 4. The potential for continued disease transmission. The clinical symptoms will suggest the type of agent involved and initial laboratory specifications may have already identified the agent. Initial cases also may provide important clues to identify a common source, groups or cohorts of people exposed and the areas to target. In the early stages of an outbreak investigation, it may be useful to survey the major providers of acute or emergency medical care serving the likely where the ill persons are located. Geared with this knowledge, we are ready for planning the investigation. B. Planning the investigation. Now, how to go about planning the investigation procedure? First of all, the method of investigation needs to be decided early in the investigation. The cohort method is often used in food-borne outbreak investigation in which the whole group of exposed individuals is known, for example, all persons attending a wedding party at which a food-borne outbreak occurs constitute the cohort of persons at risk of illness. The investigation will be carried out by questioning all cohort members about illness and food consumption. In case a cohort is too large, a random sample can be interviewed. We have talked about one of the methods of investigation, i.e., questionnaire. Let us have a look at what this method is and how it is used. As you may already know, questionnaire is a set of questions, both open-ended and close-ended, used to elicit information about any topic. It can be administered over A large sample which should be literate or semi-literate. What kind of information can be collected through the questionnaire? Can you suggest? Well, the information that needs to be collected can be listed as 1. Demographic information. This includes name, address, age, sex of the both affected and normal individuals. 2. Symptoms. These include diarrhea, fever, abdominal pain, nausea, vomiting. Time of onset and sequence of onset of symptoms. 3. Food history. The standard practice in food-borne outbreaks is to take a detailed history of foods consumed by affected persons over the 72 hours prior to the onset of symptoms. It is important to include water or other liquids consumed, as they also can be the vehicles for infection. It is also important to quantitate the intake, i.e., number of servings, glasses, etc. 4. Data screening. Filled up questionnaires need to be verified for the discrepancies, if any, while filling up of the information. As can be seen, depending on the information required, the type of questionnaire will vary. Also, the type of questionnaire used will depend on the population and the method of administration. Once the data has been collected, the next step is analysis. C. Epidemiological analysis. Once the questionnaire has been checked, data needs to be analyzed to describe the outbreak in terms of time, place, persons, symptoms, and the likely causative agent. This is how it is to be done. Time. The typical way the time course of an outbreak is to be known is to use date time of onset of symptoms and draw a histogram of number of cases by time of onset of the symptoms. The pattern displayed by the histogram will indicate whether an outbreak occurred from a single point or is it a propagation. A sharp peak in the histogram suggests that the incubation period is very short. Place. Examining the attack rates, i.e., number of persons affected total group in various geographical subgroups of the outbreak can help to concentrate on a particular area where the investigations need to be done. Persons. The personal characteristics of cases like age and sex will help in identifying the source of an outbreak. For example, if the affected persons are children, it may provide a clue to suspect a certain food. Other important information. Additional information such as place, time, group of people involved, involvement of animals which might have consumed the incriminating food, etc., will also need to be collected. After the simple analysis, it is time to determine association between different factors investigated in the outbreak. Let us see how this is done. D. Analytical epidemiological analysis. Analytical epidemiological studies seek to determine associations between certain food items and illness. The selection of a control group is a vital step in the analytical epidemiological studies. In an investigation of a food-borne disease among a cohort, the food consumption histories of all persons in attendance are collected and examined for association of eating certain food items with illness. Let us see how this is done. We will briefly go through one of the methods used to identify implicated food items by analytical epidemiological analysis. This method is traditional approach. Traditional approach. Questionnaire responses from the affected and unaffected persons are used to generate a food-specific attack rate table. For example, if 100 people attended a marriage party and 55 became ill after 4 hours, Staphylococcus aureus was detected in the stool samples of several people who attended the marriage party. The food-specific attack Rate for two food items is shown in Table 5.1. Table 5.1, Food-Specific Attack Rate Marriage Party. Look at the screen. As seen in Table 5.1, the percent of illness among persons eating and not eating each food item is calculated and compared. The kheer has the highest attack rate among consumers and lowest among non-consumers. Highly significant correlation, p. 0.001, is found with kheer. Same data can be analyzed by another method called relative risk. Relative risk is calculated by dividing the rate of illness in food consumers by the rate of illness in persons not consuming the food. Similarly, there are many methods to analyze the data like case control analysis and stratified analysis. After the epidemiological analysis, it is imperative to carry out the laboratory analysis to identify the etiological agent causing the disease. We will learn about this aspect next under the heading Collection of Samples. E. Collection of samples. Most important aspect of food-borne disease would be to collect the right sample. Now, what procedure to follow while collecting the specimen? Let us try to understand. A few steps involved in the collection of the food specimen. Collect the specimens of all leftover foods. It is often observed that once a food-borne disease occurs, all the suspected food is discarded deliberately or in the natural course. Efforts should be made to trace the sample. Normally, in the Indian context, in case of food-borne disease outbreaks, the first to investigate are police under Section IPC 304A. Often, the incriminating sample is destroyed to erase any trace of evidence. If foods from commercial sources are suspect, carefully record the label information. If the food handler's role is suspected, the hand washings or swabs of infected wounds should be collected. All the samples should be collected in sterile containers, sterile jar, beakers, or plastic jug, and should be transported to the laboratory in an ice bucket and analyzed at the earliest. Collection of biological samples of affected persons such as urine, blood is also necessary. After sample collection, the next step is analysis. F. Laboratory analysis. Laboratory analysis is the next crucial step in determining the agent which led to the outbreak of a disease. The most important consideration here is that the laboratory should be informed in advance so that all the necessary arrangements are made for analysis as soon as the samples are brought to the laboratory. Appropriate analysis should be carried out to find out the etiological agent. Whenever positive results are found, the cultures and specimens should be preserved for further confirmation. After analysis, it is time for preparing the report, as highlighted in the next step. G. Preparation of the final report. Synthesis of epidemiological and laboratory analysis data leads to identification of the causative agent, vehicle of transmission, and circumstances under which a food-borne disease occurred. A final report has to be written meticulously, documenting all the compartments of epidemiological and laboratory analysis data, with conclusions and recommendations. So you see, this is how we report and investigate a food-borne outbreak. We hope you would use this information too while reporting and investigating an outbreak in your region. 5.9 Summary. In this unit, we studied about food-borne diseases categorized as food infections, intoxications, and toxic infections. We learnt in details about important food diseases caused by bacteria, viruses, parasites, mycotoxins, and naturally occurring toxins in food. The main focus here was on the characteristics of the disease, its occurrence, etiology, incubation symptoms, mode of transmission, and preventive measures. Finally, methods of reporting and field investigation of foodborne disease outbreaks were also dealt with. Thank you for watching. We will see you in the next video.